How effective has GDNF been in clinical trials? When it comes to glial cell line-derived neurotrophic factor, or GDNF, many wonder how effective it has been in clinical trials for Parkinson's disease. GDNF is a protein that plays a crucial role in supporting the survival and growth of dopamine-producing brain cells. These cells are progressively lost in Parkinson's disease, making GDNF a focus for potential treatments. Since its discovery in 1993, researchers have conducted numerous studies to explore its effectiveness. Early clinical trials showed some promising results. In small studies, GDNF therapy led to significant improvements in motor function. For instance, one trial reported a 39% increase in motor abilities when patients were off their medications. Some patients even experienced sustained benefits for up to 2 years after treatment ended. In one notable case, post-mortem analysis revealed regeneration of dopamine fibers in the brain, which is a hopeful sign. However, not all studies have been as encouraging. Some trials indicated that the benefits of GDNF might diminish after stopping the treatment. In a phase I trial where GDNF was delivered to just one side of the brain, motor improvements were observed during treatment. But within 9 to 12 months after stopping, patients returned to their baseline motor function. Additionally, some patients developed antibodies against GDNF, which could potentially impact the treatment's effectiveness. A larger randomized clinical trial utilized a new delivery method to infuse GDNF directly into the putamen, a brain region affected by Parkinson's. This method was found to be safe and well-tolerated, with high compliance among participants. Most individuals experienced meaningful clinical improvements. However, the trial had design limitations, such as a lack of double-blind placebo control in the final stages, making the evidence less conclusive. Researchers suggested that longer treatment durations, higher doses, or earlier interventions might yield clearer benefits. Recent advancements in gene therapy approaches delivering GDNF have also shown promise. One Phase 1b study indicated that GDNF gene therapy was well tolerated, with no serious adverse effects. Participants experienced stability or improvement in motor function over an 18-month period. These findings support the need for further investigation into GDNF gene therapy as a potential disease-modifying treatment. Despite these encouraging signs, GDNF has not yet been established as an approved treatment for Parkinson's disease. Challenges remain in optimizing delivery methods, determining the right dosage, and selecting appropriate patients to maximize benefits. Ongoing and future larger-scale, well-controlled trials are essential to clarify GDNF's true clinical efficacy and long-term impact. In summary, GDNF has shown biological activity and some clinical benefits in trials, particularly in improving motor symptoms and possibly promoting dopamine neuron regeneration. However, the evidence remains inconclusive due to trial design limitations and variability in patient responses. Continued research is crucial to better understand its role as a neurorestorative therapy. Patients are encouraged to consult healthcare professionals and relying on approved treatments while GDNF remains an experimental approach.